debate today, so we're going to move straight to Lauren Pittaccio from Queen's University Belfast, who is giving another flash talk titled Modelling a Biologically Representative Environment. Does respiratory mimicking media influence the evolution of meropenem resistance in Pseudomonas aeruginosa? Thank you very much.
Yes, so hi, my name's Lauren, and I'm going to be talking about this part of my PhD, which is modelling a biologically representative environment, and does respiratory mimicking media influence the evolution of meropenem resistance in Pseudomonas aeruginosa? So AMR is becoming an increasing threat to the ability to treat bacterial infections, and we commonly use evolution experiments to control and monitor evolution throughout in a lab setting. Conventional evolution experiments usually use one single culture of bacteria and its increased antibiotic resistance in rich laboratory media, which, while it has helped us develop a fundamental understanding of bacterial evolution, it doesn't represent the complexity of a biological environment where infections occur. So to address this gap, we've been using a infection niche media modelled on the respiratory tract, specifically respiratory mimicking media, which is modelled on acute lung infection environments like pneumonia. So the key difference between respiratory mimicking media and the other lab-based media that I use, TSB, is that respiratory mimicking media contains substances that both promote and repress bacterial growth. These are lysozymes and lactoferrins, whereas TSB contains substances that only promote bacterial growth. And that's why it's used commonly in culturing bacteria within the lab. So then the next stage was whether the use of this respiratory mimicking media impacts the evolution of meropenem resistance in Pseudomonas aeruginosa. To investigate this, I did a 9-day adaptive evolution experiment using Pseudomonas aeruginosa PA01. And I, over the 9 days, exposed these bacteria to 4 different conditions: respiratory mimicking media and meropenem, TSB and meropenem, and then both medias had a control as well. The isolates that I achieved from these evolution experiments then went through growth curve analysis, MIC assays, and sequencing. So to look at the population size throughout the evolution experiment, I took an optical density reading after each daily passage and found that overall growth was suppressed in the respiratory mimicking media as increased meropenem concentrations happened throughout each day, whereas in TSB, Pseudomonas aeruginosa had a lot more continuous, constant growth. I then took the isolates at the end of the evolution experiment and use an optical density reading to establish whether these counted as they survived the evolution or didn't survive. And I found from that meropenem resistance vulnerability was reduced in respiratory mimicking media, as 8 out of 10 of the isolates in respiratory mimicking media and meropenem survived, whereas 10 out of 10 in all other conditions survived the evolution experiment. I also did MIC assays, which are not pictured here but are on my poster, that proved that meropenem resistance was evolved by comparing the MIC of the media plus meropenem, and then the respective control sample found that the MIC was much higher in the meropenem resistant colonies. Most recently have sent 5 of each of the condition samples off for whole genome sequencing to see if there were any specific genes that were different between the 2 medias. In both medias, I found that there was the OPRD gene, which is a meropenem associated resistance gene throughout, but the rest of them did not have a the other mutations between the 2 medias were very different, and there weren't any other similarities between the 2. So overall, to sum it up, I found that meropenem resistance was evolved in P. aeruginosa in both medias. However, meropenem resistance evolvability was reduced in respiratory mimicking media. And if you'd like to know more, I have a poster on board 16, and I'll be around the poster in the session. Thank you.
Great, thank you very much, Lauren.